welcome to the first session of Oceans Alive. It doesn't feel very spring-like tonight, but this is always an excellent way to welcome spring with the recipients of the local science fairs from Falmouth Academy and Falmouth High School. And I always think this is the most exciting seminar that we present in the Oceans Alive series because it's bright, enthusiastic young scientists who've really paved the way for the students that follow in their footsteps and also makes a mark even if they don't continue in a scientific profession. It shows the talent and creativity and insight that the Falmouth students have brought to bear and brought into this auditorium. So the four speakers that we have this evening have excellent projects. They've received great honors for their projects at their schools, and they're going to share their results with us this evening. They've taken polls as who's going to go where, and the first one this evening will be Don Swanbeck. And Don is a junior at Falmouth Academy. He won top honors for his project at Falmouth Academy Science Fair, and he will be telling us about his work on the effect of heavy metals on the growth of phytoplankton. Don?
Hello. Hi, I'm Don. Is this thing turned on? I just don't know. I'm sorry. Oh, okay. For my project, well, last year I had studied the effect of temperature on the growth of 5 species of phytoplankton, 2 dinoflagellates, 2 diatoms, and 2 dinoflagellates, 2 flagellates, and a diatom. And as I'm sure you probably know, phytoplankton are kind of the base of the aquatic food chain. So anything that happens to them is either directly or indirectly hurting the levels that feed off of them. So it's very important to know what affects these creatures and what pollutants might hurt their populations. I guess the pollutant that I studied this year was heavy metals, and I chose lead and copper as heavy metals to test them. And some trace amounts of these metals exist already in the background, but they're also introduced into the environment from industrial sources. Copper comes from the bottoms of boats from anti-fouling paint. Copper sulfate is used as an algicide. Lead comes from lead iodide on the highways and industrial, and it really can hurt these populations. Either, depending on the species, it can either damage them slightly or not at all, or can completely wipe out a population. And this has happened before, so it's pretty important to know how these heavy metals affect them. And the species I used were Procentromycans and Heterocapsa triquetra, the 2 dinoflagellates, T. Isochrysis galbana and Rhodomonas salina, 2 flagellates, and Thalassioceras viceflogi, a diatom. And I wanted to determine how lead and copper affected these species, I used growth based on chlorophyll increase to determine how they were affected by the heavy metal and how it interfered with their metabolic process. So, I hypothesized that as the concentration of heavy metal in the environment, both copper and lead, increased, their growth rates would decrease constantly. So what I did was I inoculated F/2 media and test tube racks, one for each species with the phytoplankton species, same amount for everything. And I introduced small amounts of lead chloride and copper sulfate into the solutions to form concentrations for lead of 10, 20, 50, and 100 micrograms per liter, and for copper, 10, 20, 50, 100, and 200 micrograms per liter because it's a less toxic heavy metal, and I used a control as well. And there were 3 replicates for each substance. And I placed these setups in all in one room that was 15 to 18 degrees Celsius, and randomized the tubes about every other day when I checked on them to see what their fluorescence was. And I did that using a fluorometer. And once I had got that, I took the averages from the replicates, and I would plot their average growth rate based on concentration. And here, for example, is just one. It ran for about 35 days, but I guess I only ended up analyzing the first curve because I was interested in their growth rather than their death. So as you can see, the circle kind of represents, it's kind of confusing in this particular one, but that kind of gives an idea of what the growth curves would look like typically. So so I would graph them like that, and in addition to that, I would use a growth equation. Can't do that. I'd use a logarithmic growth rate equation to determine the growth rate per species based on the first 5 to 7 days, and I would rank them in decimal form and try and get an idea of which concentration would affect it. As you can see, for procentromycans, there doesn't seem to be any clear pattern to the ranking as the concentration increases. So I wasn't able to draw any significant conclusions from that one. But for Rhodomonas salina, I had to omit some because of small amounts of contaminants that were found in a replicate. So rather than risk error, I took out the points. And it kind of shows that as the concentration increased, the logarithmic growth rate would also would decrease, showing that the smaller the concentration of lead, the better the growth rate. And over here, it follows sort of the same pattern, even though number 10 doesn't seem to fit in with the rest. The 10 micrograms per liter concentration didn't seem to fit, so I can only assume that that's a fluke. T-iso didn't seem to make any sense in this one, so I didn't see any real pattern how lead affected it. In this one, here's an example of kind of an interesting thing, that the copper actually seemed to help it, and copper, more than lead, actually is needed in small amounts of metabolic processes, so small amounts of copper actually help the growth rather than inhibit it. And as the concentration then increased from that, the growth rate would begin to decrease. And that same pattern actually showed up in Heterocapsa for both lead and copper. Not necessarily surprising for copper, but a little bit surprising for lead that it would go 3, 1, 2, 4, 5, just because lead, to my knowledge, doesn't seem to be essential for life or actually help it in any way. For Thalassiocera vice-flogi, I couldn't detect a real pattern to that, and for the lead half and Thalassiocera with copper, I determined that copper negatively affected it, even though the 20 and 50 concentration seemed to reverse. The general pattern indicated that as the concentration of copper in the environment increased, the growth rate would decrease. Kind of an example of that is the effect of lead on Heterocapsa. As you can see, for Heterocapsa, I wasn't able to measure it for the length of time that I was Procentrum, but it was sufficient to measure just this section of the graph because I was only analyzing the growth rate. But as you can see, the 10 micrograms per liter concentration of lead actually seemed to be better than the control setup, and it just seems to, just kind of shows that lead in some cases would actually help it. And analyzing the data with variance and range statistics, I was able to form a conclusion chart where I was able to determine basically how lead and/or copper affected the species of phytoplankton, and some of the more interesting ones that are visible are the effect of copper on Thalassioceras and the effect of copper and lead on Heterocapsa, because in Heterocapsa's case, lead, I believe, helped to stimulate growth in that case, which seemed to be a little bit odd. So I guess in conclusion, my hypothesis wasn't really supported because I thought that as the concentration increased, the growth rate would decrease all the time for every species and for every type of heavy metal. But in some cases, the phytoplankton didn't seem to be affected by the heavy metal in the environment. In other cases, it was actually helped by small amounts of the heavy metal, and in still other cases, it actually did act like I hypothesized. And there were very, very small amounts of error in the experiment because I used automatic pipettes and had autoclave the solutions to prevent infestation of contaminants, and as such, phytoplankton respond differently depending on species to each respective type of heavy metal. Any questions? Take that on.
We have time for a few questions for Don.
Did you consider the possibility that there may be a different effect due to complexation of the metals, the heavy metals, with regard to the different species? And in one instance, you may have got more free metal, and in another instance, you might have got more complex metal, which would indicate differences in toxicity.
I'm not quite sure I understand the difference between complex and...
You use Peppo with two media,
Uh-huh.
and you added heavy metals to that,
Mm-hmm.
and you drew phytoplankton. Now, in the presence of extracellular organic material, in some instances, there could have been a complexation of the free metal, which could change its toxicity.
I guess I wasn't aware of that, so I guess that's a possible source of error.
It's one possibility, I think, that you might consider. I don't know how that would affect the results, but
I could look into that and see how it may have affected my data. Any other questions?
That's some very interesting results. There's been some examples in toxicity tests with fish in which low levels of the toxicant actually improves the performance of the test animal or the test species. So it seems like the results are consistent with that kind of process.
I might want to try it with other levels later in my life just to see how it affects some of the other levels of the marine web. So I'm just curious. Did that occur because the fish adapted to the heavy metal, or because it actually helped it?
The hypothesis is that the toxicant actually stimulates the immune response, at least in animals, and that actually mobilize enzyme systems and so forth, so they actually perform better, grow faster, eat harder, things like that. So under this low level, it's
hurts them.
Degradation of soil nature. So I think your results are consistent with that kind of.
Yeah, I think so.
It's been observed in a lot of different marine and bird groups as well, and it's a process called hormesis.
Hormesis?
Hormesis. So it's, and copper is one of the compounds that certainly does stimulate growth rate in crab larvae. So look up that $1 million word. But it is a very interesting phenomenon that occurs throughout the animal kingdom and in plants as well, that, you know, a little bit of toxicity enhances growth and survival.
It's interesting.
And nobody really knows all of the mechanisms involved, so. There's a buzzword you could use. Any other questions for Don?
Okay,
Nice job.
thank you.
As our next speaker gets wired for sound, to introduce Jill Morosky, who is a junior at Falmouth High School. And Jill captured the second prize at Falmouth High School for her project titled Effects of Tropospheric Ozone on Phaseolus vulgaris. Jill?
Hi, I'm Jill Morosky. My project this year is called The Effects of Tropospheric Ozone on Phaseolus vulgaris, which is the common green bean plant. Ozone is a very unstable and highly reactive gas in the atmosphere. Each molecule of ozone is made up of 3 oxygen molecules. That's what makes it very unstable. And there are 2 types of ozone that can be found in the atmosphere. First is stratospheric ozone. The stratosphere is the second layer in the Earth's atmosphere. It's about 5 to 8 miles into the atmosphere. And you've probably heard that the ozone layer, which is beneficial to life on Earth because it blocks out harmful ultraviolet rays. This is found in the stratosphere. There's also tropospheric ozone. Troposphere is the ground level up to 5 miles. This can be bad for life on Earth because it can bond to the life and damage it. You can see the different levels of ozone around our state in New England. You can see that this is a fairly good level of ozone around the Cape, maybe because of the winds that are around the water. And you can see near urban areas, there's a lot more ozone because of the industry there. And so the way I got the idea for this project is we had summer homework for AP biology about the ecology and the environment, and I learned about ozone, and I knew that was what I wanted to do for my project. I had a lot of trouble actually acquiring the ozone that I was going to use for this project because it has a very low half-life. It disintegrates in about half an hour. That's its half-life, and so I needed a way to actually make the ozone myself, so that I could actually use it in this project. And so what I used to make the ozone is an ozone generator. This is commercially used as an air purifier. It bonds to the particles in the air, and then it oxidizes them, and that's the way it cleans the air. So the first thing I did was I germinated bean plants in December, and I waited until they're mature plants. And the first thing I did was test the photosynthetic rates of these plants. My hypothesis was if I expose these green bean plants to ozone, that they would have both a lower photosynthetic rate and a lot of physical damage done to the plants. So what I did to determine the photosynthetic rates of the plants was I got a bell jar, and I put the plants under the bell jar with a CO2 meter. And for 10 minutes, I tested the amount of CO2 reduction that was taking place in the bell jar. Since in photosynthesis, plants use up CO2. This has a little fan in there to circulate the air, and I used a CBL system, which I got at the high school, to do this. This was before I did any ozone testing, so I could compare before and after ozone testing. Then what I did was actually tested, I tested the plants. I put them in an exposure chamber. This was connected to the ozone generator, which would be around there. This is a picture. I put the plants in a Tupperware container, and I sealed the ends. And these are dryer hoses that connected, one end connected to the ozone generator and into the exposure chamber, and then this led to the outside where the ozone couldn't harm anyone. I had to be very careful doing this because ozone can be potentially harmful to people, so I had to wear a mask and goggles when I was doing this. And the way I actually measured how much ozone I was testing on my plants, I had a control, a low, medium, and high level. This is an ozone sonde. I got it from Woods Hole Oceanographic. This tests the amount of ozone in the air by, it has positive and negative potassium iodide, and in the presence of ozone, this creates a electrochemical current, and then you can measure the amperes of the electrochemical current, and then can plug it into a formula, and that can tell you how much ozone's in the air. And the levels of ozone that I actually tested were at the normal amount of ozone that's in the air, which I tested, was only 3.6 parts per billion, which is very low. And then on my ozone generator, there was a low, medium, and high level. The low was at 360 parts per billion. The medium was at 615 parts per billion. And the high level of ozone was 930 parts per billion, which is almost a part per million, which is a lot of ozone exposure. And so I tested my 3 levels of plants for 12 hours in the exposure chamber. And after that, I looked at the effects of the visual and physical effects of ozone on the bean plants. You can see that this is a control plant, has almost no damage at all because I didn't actually expose it to any ozone. And then you can see this is a high level plant. You can see there's a lot of flecking on the leaf surface. I think this is because the ozone bonds to the surface of the plant, and then it actually takes off part of the chlorophyll of the plant that causes this flecking here. I also studied this in greater detail. I took the plants to MBL, and I had photomicrographs taken. You can see this is these are control plants, and these are the high level plants. You can see at one time you can see this is the control plant, the underside of the surface. You can see almost no damage. And then you can see high level plant, there's a lot of the ozone took off a lot of the chlorophyll of the plant. You can actually start to see the individual cells here. You can see that there's a lot of clear area here where the ozone took away some chlorophyll. And then you can see the stomates, which are the mechanisms that open and close to regulate the gas exchange in the plant. I actually have a greater detail of that. You can see this is a lot more green than this is, because it took away the chlorophyll or some of the cells. And this is a stomata, or stomate, and it has 2 guard cells surrounding it, and these are subsidiary cells which surround the stomate. You can see this is closed. They are open in the daytime and they close during the nighttime. And you can see that the cells around here are damaged in the high-level plant. This is at 0.015 millimeters right here. You see this is open. I think what happened with the ozone is that it damaged, the stomates require water to open and close, and I think the ozone interfered with the plant's ability to take up water and actually shrunk some of the cells. And so it sort of, I think it froze the stomata in place right where they were, so they couldn't open or close. You can see this one might be working just normally. And so the second thing I looked at was the actual photosynthetic rate of the plant. And I tested the plants again in the bell jar. You can see these are the control plants. This is before they were tested to ozone. This is one day after they were exposed to ozone, and this is 1 week after they were exposed to ozone. You can see that the control level varied a little bit. But you can see with the low, medium, and high level of ozone that they declined in photosynthetic rate much more than the control in before they were subjected to ozone. Another thing I looked at were the leaves of the plants. I measured the tips of each leaf on each plant before and after exposure. You can see that before and after exposure and after ozone exposure, they were measured and added up. These are the total changes in the leaf width before and after the plants were exposed. You can see that there was a lot of growth even after the plant was exposed to ozone in the control, and a little bit lower rate with the low plant. And with the medium plant, it grew 6.2% more. You can see with the high plant, it actually decreased in leaf width. This is also, I think, because of the interference with the water, which actually shrunk the plants a little bit. Also, I looked at the primary leaves of the plants after exposure. Primary leaves are the first leaves of the plant that sprout. They're the oldest leaves. They're heart-shaped, and they're more vulnerable since they're older. And you can see with the control, I ranked all the primary leaves as having slight, moderate, severe, or dead leaves. You can see with the control, most of the damage was only slight. You can see with the high plant, all of the primary leaves were dead. The last response variable I looked at were the number of beans that were produced and the bean length after they were exposed. You can see that this is a control plant, and it had a lot of variation with the number of beans that were produced. You can see even the average was greater of the number of beans produced. I had three replicate plants for each control group, or each treatment group. But I averaged them, and it was about 5, and you can see that is a lot greater than the average for each of the other plants. You can see that these are more extreme. They have higher and lower amounts of number of beans produced. And you can see, I think this is because the ozone inhibited the growth of these plants, and this one just kept on going, and that's why I think it has a lower amount of growth also. Then I looked at the actual length of the beans after exposure. See, also here, they're more extreme. With the control and low, they're more extreme bean lengths, and I also think this is because of the continued growth, while the growth of these ones were actually stopped. You can see that the average was pretty much the same. And so my conclusions with this project was, since there was a great deal of physical damage done to the plants, such as reduced leaf width and flecking on the front of the leaves and damage to the stomates, my first hypothesis was generally correct. Also, since the short and long-term photosynthetic rates were affected, this proves the second hypothesis to be generally correct. The length of the beans and the number of beans produced was greatly affected by ozone. And this is important because agrarian crops and agriculture will be greatly affected in the future when there's more industry in the world. The ozone that will be accumulating will affect how the physical attributes of the crops that will be growing will come out. And there are several people I'd like to thank doing this project. People from the high school and who helped me with my project. Thank you. Oh, I found that on the internet. I'll show it again. Yep. It was on the internet. I found it under ozone in the United States, in Massachusetts.
Are you aware that up on top of Mount Washington they have calculated this kind of information?
Yeah.
So there are, they do have ozone up on top of Mount Washington. So that's
Yep.
why I'm curious to see so much of the northern language is all green. That's what I
Yeah, probably because it's a little bit higher up, they have more ozone exposure there. Any more questions? Yep.
How many plants did you have in each of your groups?
I originally planted 15 plants, but I ended up only using 12. I had 4 treatment groups, the control, low, medium, and high, and I had 3 replicate plants for each group. Yep.
Is the leaf damage permanent or as you recover?
I looked at it both one day after and 1 week after, and I think it was permanent. The leaves didn't seem to be healing themselves, so I think it is permanent. Any more?
Our next speaker is Emily Stephen from Falmouth Academy. Emily is a sophomore, and she was also a first place winner at the Falmouth Academy Science Fair for her project on Vibrio fischeri. The effect of oxygen and carbon dioxide on bioluminescence. Emily?
Hello. Let's see. My project was on Vibrio fischeri, as she just said. This is kind of a bad Xerox, but it's just a picture. Vibrio fischeri is a bioluminescent bacteria, which means that it converts chemical energy to radiant energy inside the cell. Let's see. Vibrio fischeri specifically is often used in toxicity experiments, mainly because the fact that it gives off light makes it easy to test how its health is doing. And in nature, Vibrio fischeri inhabits the monocentrid gland of fish, which is right there. And it also inhabits some squid, the spilid squids, which are actually cuttlefish. So it can also live free in the ocean, but it's not, a lot of people have asked me, it's not what you see at the backside of ships when they're flowing along. Those are dinoflagellates. They give off light, mostly during the day, and the reaction that causes the light involves oxygen. This was one of the main aspects of my project because a lot of scientists have speculated that the fact that the process involves oxygen it's led them to believe that the process of making this light evolved as a method to get rid of oxygen in the early atmosphere with primitive cells. They are anaerobic, so oxygen could have been detrimental to their health, and a way of disposing of oxygen would have been evolutionarily advantageous. And what I did, the purpose of my experiment, was to test the effect of increased oxygen and carbon dioxide levels on Vibrio fischeri. I hypothesized that both oxygen and carbon dioxide would cause the luminosity of the bacteria to increase. Now that I look back on the hypothesis, I'm not convinced that it was a very educated hypothesis, but that was the hypothesis. And all right, what I did was I took, you see that? Okay. I took 16 cultures of Vibrio fischeri, which I acquired from Carolina Supply Company. I inoculated 16 cultures, and I grew them in environments that were enriched with one with 13% oxygen, which was 2 tubes of oxygen in my setup. 4 of them were enriched with 7% oxygen, which is 1 tube in my setup. 4 of them were enriched with 2 tubes of carbon dioxide, which is actually 10,000%, the normal amount in the atmosphere. And the reason is that there's so little carbon dioxide in the atmosphere to start with. There's only 0.03% carbon dioxide in the atmosphere. But the main reason that I used carbon dioxide originally was to take the place of the oxygen, because I wanted to try to use, to have setups where I had lots more oxygen, a little bit more oxygen, a control, and then less oxygen. So I had the carbon dioxide to take the place. I had 4 controls then. Now, this is the setup of the environments. It's basically a Ziploc bag, which I had sealed with duct tape on the top, and I tested it beforehand for any holes that might give out air, anything like that. And so what I did was I put either 1 or 2 gas tubes, depending on the variable that I wanted, into the bag. And then I also put in the test culture. And the way I got the oxygen and carbon dioxide, 2 very similar, simple setups. I got the carbon dioxide from dry ice, and the oxygen from a potassium iodide hydrogen peroxide solution. It was a pretty simple setup. I designed it to minimize the amount of air other than the needed gas in it. Okay, so I tested the cultures for light every 4 hours for 3 days. I used a Pasco high sensitivity light sensor, which is this one here. Let's see. Basically what I did was I passed the light sensor along the tubes to get the highest number that I possibly could. And then when I had all of the data, I sifted through all of the data points to look for the highest light intensity that I had gotten from the light sensor. Okay, and the results that I got, here is the original graph. This is the averages. Remember, I had 4 replicates for each variable. The carbon dioxide are the circles. The oxygen ones are triangles, either right side up or upside down, depending on how much oxygen were in them. And the control is the diamond. Basically, when I looked at this graph for the first time, it's sort of, the first impression that I got was there's no data. The error bars on here are standard error, and most all of them overlap, which is kind of discouraging. But what I did find were there were some points here, 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 and here on the carbon dioxide where they don't overlap. The error bars don't overlap. And I admit that that's not very convincing until you look at a different graph that I made, which is the same data, but instead of just giving the total light intensity, what I did was I found the difference from the control. And if you look on it, the carbon dioxide, these are the significant points in here. Even though you know that most of the carbon dioxide points aren't statistically significant, if you look at the trend, nearly all of the carbon dioxide points, except for here, 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 yeah, and that's it, and there's one that's exactly the same as the control, they all follow the trend of being less luminous than the control, which suggested to me that there was something about the carbon dioxide that was causing them to glow less brightly. The idea that I originally had, that it was taking away the oxygen, didn't really convince me because all of the oxygen examples, I'll show you the other one again, they didn't really show a statistically significant difference from the control. All of the error bars overlapped, so it didn't really convince me. I guess what the idea that I came up with was that there must be something specific to carbon dioxide that's causing them to glow less brightly. That could be that carbon dioxide's mildly toxic to the bacteria, that it inhibits the chemical reaction, or, which is kind of likely, that the carbon dioxide was interacting with the water in the agar to make it more acidic, which would definitely have influenced the luminosity or the health of the bacteria. If I were to do this again in the future, I'd use different levels of carbon dioxide. And if possible, I would try to test the acidity of the agar throughout the experiment. Unfortunately, that would be really hard because as the experiment went along, my main goal was to maintain the seal so that I didn't let any carbon dioxide or oxygen escape from the Ziploc bag. So my hypothesis that oxygen would cause the bacteria to glow more brightly was not supported because the error bars mostly overlapped. There wasn't a significant trend. And my hypothesis that carbon dioxide also would cause the bacteria to glow more brightly was not supported because the trend was that they were less bright than the control. I guess that's pretty much it. Are there any questions?
Questions for Emily?
Questions? Yes?
Was the age of your cells comparable in each instance?
Well, the actual cells, it would be difficult to tell, but all of when I inoculated the bacteria, they were all originally inoculated from the same culture. So I assumed that the bacteria in the one culture would be about the same. And then they were all inoculated at the same time so that they would have comparable ages.
How old was your original culture?
It was about 2 days old. Any more questions? All right.
Our final speaker this evening is Joe Rago, who won the first place award at Falmouth High School Science Fair. And Joe is a junior, and his project this year is investigation, isolation, and identification of an allelopathic agent.
All right. Good evening. My name is Joe Rago, and as she said, my project is the investigation, isolation, and identification of an allelopathic agent. Before I get started, I'd like to thank some people. I'd like to thank Dr. Johnson, my dad, Dr. Rago, and Dr. Jeff Seawald. All right. What is allelopathy? Specifically, allelopathy refers to any process in which a plant, microorganism, virus, or fungi produces a secondary metabolite, a chemical, which influences the growth and development of an agricultural or biological system. Allelopathy is primarily a form of natural selection where an allelopathic species, by eliminating other plants or other organisms in the same environment, has a leg up, so to speak, on competition for resources and other nutrients. All antibiotics are allelopathic in nature. Penicillin is an allelopathic species. But what I sought to do was to identify an allelopathic plant. So I sought to determine if the germination and early growth of the radish plant could affect the simultaneous growth and germination of the lettuce plant. I wanted to eliminate, or at least significantly reduce, resource competition as a cause of the observed effects, and then to extract the allelopathic agent and then to identify it. So what I did, I had a varying level of radish seeds in combination with a constant density of lettuce seeds. Here I have a schematic diagram, so to speak. So for each replicate dish, the number of lettuce seeds stayed constant. It was 10 lettuce seeds to a dish. But as the treatments continued, there was an increased number of radish seeds. So although the lettuce seed remains constant here, it becomes proportionally less of the total seed population. In addition, there were also a series of control replicate dishes for each total seed density. For example, here for the third treatment, there were 10 lettuce seeds and 25 radish seeds. So there was an analogous control of 35 lettuce seeds and a separate series of analogous controls with 35 radish seeds. And by comparing the results of the control dishes by species to the results of the treatment dishes by species, you could see whatever hypothetical effect there might be would be demonstrated in the difference between the control and the treatment plants. Just to go into the preparation a little bit, the seedlings were all grown in absorbent felt pads contained within Petri dishes. These felt pads were highly absorbent, so there was plenty of water for the plants to grow during the course of the experiment. The dishes were not opened during the experiment, and the addition of water was unnecessary. They were all grown under constant conditions. There was constant light, certainly constant temperature, and constant exposure to gaseous nutrients. At the end of the experimental period, which was 10 days, all the plants were measured to the nearest millimeter. So here you can see the results for the radish plants. The blue triangle here is the length of the treatment radish seeds, those grown with the lettuce, and the red dot is the length of the control radish seedlings grown at the same seed density but in separate dishes. And you can see in all cases, except for the one anomaly here, the third treatment, the results were identical, statistically identical. If I could just have it here. All results were analyzed using confidence interval, which is a kind of derivative from standard deviation. It takes into account not only the variation within the data set, but also the number of observations to draw a more accurate inference about the mean. Now here, if you look for the lettuce plant, you'll see there is a statistically significant reduction in the growth of the lettuce plant as the radish seed density increases. For the lower treatments where there's either an equal number of radish and lettuce seeds or the lettuce seeds outnumber them, there's not a statistically significant difference. But as the seed density increases here, there is a large difference in the statistical difference between the control and the treatment plants. One of the problems many researchers have had in studying allelopathy has been separating resource competition from an actual allelopathic effect. That is, separating maybe the plants competing for the same water, the same nutrients, the same light from an actual release of an allelopathic chemical into the environment. My experimental design did a number of things to reduce the presence of these competitive factors and test more accurately for an allelopathic process. It investigated only the early growth, the germination and the early growth of the plants. As they grow, there's an increased complexity of analysis of results as they grow lateral stems or more developed leaves, and they start to need soil. One of the other benefits of this design was that it eliminated soil from the equation, so to speak. Soil has varying concentration of nutrients, varying presence of microorganisms within the soil, and so eliminating these factors allowed greater testing of the allelopathic effects of the radish plant. The pads, as I said before, were absorbent enough to hold enough water so that the plants were not directly in competition for that water. There was certainly enough there for each plant to grow on its own without competition. And there was plenty of light and gaseous nutrients available. So based on that data, it was shown that the radish seedlings were not statistically significantly different from each other. There was the one anomaly, but that's certainly to be expected, I would say, and not particularly significant. And there was, in addition, there seemed to be an allelopathic effect on the lettuce. It seemed that there was something in those systems, which was causing a reduction in the lettuce growth. However, it was not conclusive. If I pull that up again, there is a difference, but not a conclusive difference. So the reason I felt for this was because the radish plant growing in the system had to release that toxin, allelotoxin, into the environment. As they're both growing simultaneously, there is sort of a grace period there where there is sections, periods of time where there is no allelotoxin in the system to affect the lettuce plant. So what I wanted to do next was to grow the radish seeds before, remove them, and then grow the lettuce plants in the same dish so that the hypothetical allelopathic agent is already in that system from the moment of lettuce germination. So an increasing density of radish seeds were germinated in the dishes, and at the end of the 10-day period were removed. All the radish biomass was removed, all the stems, the roots, any lateral roots or root hairs that might have grown, the seed coat was removed. And then the lettuce seeds were sown into those same dishes. The pads were still moist enough to allow the lettuce plant to grow effectively. And as you can see here, here's the control dishes. It was around 5 or 6 centimeters for those. These are again analyzed using the confidence intervals. And as the radish seed density increased, the growth of the lettuce significantly decreased. In the last 2 cases, when there were 50 radish seeds to the dish before the lettuce growth, or 75 seeds per dish before the lettuce growth, growth was held to under 1 centimeter, which is particularly significant, and certainly for all these treatments, a statistically, very highly statistically significant reduction in the lettuce growth. So competition was not a factor in the design, as I explained. This experiment also showed that the hypothetical allelotoxin was water-soluble because it was in that system and absorbed by the lettuce plant. It was shown that the most probable mode of phytotoxicity was through the water supply. The first experiment and the second experiment, in conjunction with each other, provided strong, very strong evidence that the radish did indeed produce an allelopathic agent. At that point, I wanted to extract the allelochemical. I used deionized water as a solvent and extracted it from differing masses of radish seeds, both whole and crushed, both whole and ground, and from radish plants which had been grown for 10 days. The differing solutions were soaked in the deionized water, agitated with a magnetic stir, and at the end of the 24-hour period were filtered. And all solutions which I extracted from the radish plant were shown to be completely inhibitory to the growth of the lettuce. Those solutions were applied back into the systems, and the lettuce was grown using those solutions as the water for its germination. And in all of the 5 solutions, it was completely inhibitory to the growth of the lettuce, which constitutes very strong evidence that there is indeed something in the radish plant which causes a reduction in growth of or in this case, a complete inhibition of growth of the lettuce plant. Certainly these solutions are in higher concentration than the plant produces naturally, but it is a very striking effect when you see here that there was no germination at all. It was completely inhibitory. At that point, I wanted to to identify the allelochemical which I believed I had isolated. I used mass spectroscopy and gas chromatography. Gas chromatography is a technique to separate volatile organic compounds based on the different partitioning behavior in relation to their boiling points. They're separated due to differences between a mobile gas phase, which flows through the gas chromatograph, and their actual gaseous ions. Because there's differing boiling points hypothetically within your sample, the gas chromatograph is started. It's a series of coils which is contained within an oven, and the temperature is gradually increased as the analysis proceeds. Mass spectrometry analyzes differences in the mass to charge ratios of ionized atoms. Because compounds have distinctive fragmentation patterns, by comparing the fragmentation of the compounds in your sample to the predicted fragmentation, you're able to give an identification of what it is in your sample. So a mass spectrometer creates gas phase ions. It separates the ions in time or space, and then analyzes the mass to charge ratio of those ions. And I have here one of the mass spectrographs, and you can see this is identifying the chemicals that were indirectly injected into the GCMS. And there's a distinctive peak whenever there is a chemical in concentration in the system enough to be identified. For each peak on the mass spectrograph, there is the fragmentation patterns for that peak. Here is carbonyl sulfide, which has the formula COS, carbon, oxygen, and sulfur. Because as it fragments in the GCMS, well, for example, you have the oxygen, which would break off. Oxygen is diatomic, meaning it bonds to itself, and oxygen has a molecular weight of 16 grams. So you get a distinctive peak here at double 16 at 32. So the compounds which I identified were all low-weight organic, volatile organic compounds, carbonyl sulfide, acetaldehyde, acetone, dimethyl disulfide, and ethanol. If you see here on the mass spectrograph, they were in fairly low concentrations. I had them stored in an Erlenmeyer flask as opposed to a Teflon-coated test tube or something of that nature. The organic compounds will dissipate out of solution over time, and that certainly resulted in lower concentrations of these chemicals. And at this point is where I ran out of time. But I would have to, in order to conclusively identify which of these chemicals are compose the allelopathic agent, they would have to be applied back to the lettuce in differing concentrations and differing combinations to conclusively identify which chemicals are indeed causing the allelopathic effect on the lettuce. So, are there any questions for anyone? Mr. Stevens?
When you eat a radish or eat lettuce, you actually think the radish has a stronger taste than might be bad.
Mm-hmm.
Makes sense to check and see if the lettuce has any allelopathic.
Yeah, afterwards, that was something that I thought it might have been advantageous to look into. Perhaps the combination between anything the lettuce might secrete might react with what the radish is secreting, which might be causing those effects. I really didn't look into that, but that's certainly an area that it would be good to look into in the future.